Hi and welcome back to Free Science Lessons. By the end of this video, you should be able to compare two different models for how enzymes function. These are called the lock and key model and the induced fit model. In the last video, we saw that enzymes are globular proteins. On the surface, the tertiary structure of the enzyme folds into a three-dimensional shape called the active site. The active site is the part of the enzyme where the substrate molecule attaches to form the enzyme-substrate complex. As we've seen, the tertiary structure of the active site is specific to the structure of the substrate molecule, and this explains why enzymes are specific. Temporary bonds form between the substrate molecule and the amino acids on the surface of the active site. These temporary bonds help to lower the activation energy of the reaction, and this explains how enzymes increase the reaction rate. Now, when scientists first began to look at enzymes, they thought that the tertiary structure of the active site is fixed and does not change shape. So essentially, the substrate molecule slots perfectly into the active site, a bit like a key slotting into a lock. Scientists call this the lock and key hypothesis, or the lock and key model. However, later scientists realized that this model was not correct. In fact, the tertiary structure of the active site changes as the substrate molecule approaches. As the substrate starts to form bonds with the amino acids in the active site, the tertiary structure of the enzyme adjusts, so the active site molds itself tightly around the substrate. This change in the tertiary structure of the enzyme ensures that the active site fits perfectly to the substrate, and the bonds that the substrate forms with the active site help to catalyze the reaction. Scientists call this the induced fit model, or the induced fit hypothesis. Molecules which are not the substrate cannot form the correct bonds to the correct amino acids in the active site. Because of this, the tertiary structure of the enzyme does not change, and that means that the shape of the active site does not adjust to fit the molecule. And this helps to explain why enzymes are specific for their substrate. So as you can see, the model for how enzymes function has changed. Experiments have shown that the induced fit model is correct. Okay, so hopefully now you can compare the lock and key model with the induced fit model.